This method can help answer key questions in the immunology field, such as how exactly immune cells behave in a physiologically relevant context, like how to search target cells efficiently in a three-dimensional environment. The main advantage of this method is to generate a very thin sheet of light to illuminate only the focal plane without affecting the off-plane cells. This enables a very fast acquisition speed with a pronounced reduction of bleaching and photocytotoxicity. Demonstrating this procedure will be Dr. Renping Zhao, a postdoc from my lab.
To begin, under a cell culture hood, transfer 400 microliters of chilled collagen stock solution to a sterile 1.5 milliliter tube. Slowly add 50 microliters of chilled 10x PBS. Then, mix the solution by gently tilting the tube. Add 48 microliters of 0.1 molar sodium hydroxide to the collagen solution to adjust the pH to 7.2 to 7.6. Use pH test strips to determine the pH value of the mixture. Add 2 microliters of sterile distilled deionized water to bring the final volume to 500 microliters. Mix well and store the collagen solution on ice or at 4 degrees Celsius until further use. After fluorescently labeling live